Good day and welcome to the Greens Academy. This section of biology is reserved for high school students and more precisely opposite science students. So we will expect that each time you get to this section, you identify the topics and you now receive the lessons that will be involved in each chapter. Our first chapter for this opposite science class is enzymes. Enzymes. So we are going to be talking about enzymes, and we are going to start with our first lesson. But before we get into the details, we will need to have the objectives of this chapter. It is very important for us to understand the objectives. It is only when we must have understood the objectives that we will be able to get the concept that this topic or chapter is talking about. At the end of this chapter, the students should be able to 1. Explain the properties and functions of enzymes. In this particular objective, the properties and functions of enzymes must be well understood. Therefore, at the end, the student should be able to explain the properties of enzymes as well as the functions of the enzymes. The second objective will be to describe the effects of the different factors on enzyme catalyzed reactions. Next, we may want to ask ourselves, what are enzymes? What are enzymes? It is imperative and important for us to understand the definition of enzymes. From the definition of enzymes, we will now be able to understand the objectives of this particular chapter. Enzymes are specialized proteins secreted by living cells. We say they are specialized proteins secreted by living cells. So, they can only be secreted by living cells because we may bring other aspects like dead cells, or we may ask ourselves, do dead cells secrete enzymes? So, from this first point of view, we will say that enzymes are secreted by living cells. Cells that carry out metabolic activities. And these metabolic activities can either be anabolic or catabolic. Enzymes can be defined more precisely as biological catalysts. They act as catalysts. That is a substance which speeds up the rates of chemical reaction but remain unchanged at the end of the reaction. This is very important. We have said they are biological catalysts. It means they are proteins. We have said they speed up the rate of chemical reaction but remain unchanged at the end of the reaction. This means that at the end of an enzyme catalyzed reaction, the enzyme does not undergo any structural or chemical change. Its function is to speed up the rate of reaction. Its function is to help the reaction to occur normally. They are needed in minute quantities by the cells. This is also very important because we may think that since enzymes help to speed up chemical reactions, they must be poured in large quantities. No, they are needed in minute quantities. Therefore, the amount or the quantity does not matter. It just depends on the type of reaction they are to catalyze, and that will determine the amount of enzyme needed or to be secreted to carry out that particular reaction. Therefore, the quantity needed must not be large because they can react even at minute quantities to carry out the appropriate reaction. They are biological catalysts because they are protein molecules manufactured by living cells. That is why we call them biological catalysts, because they are protein in nature and they are manufactured by living cells. I want us to understand this very well. Since we are insisting on living cells, it therefore means that dead cells may not be able to produce enzymes. But those dead cells may have microorganisms growing in them that bring about decomposition. At that point, those microorganisms may be carrying out some metabolic activities, and enzymes may be secreted in those microorganisms that will be able to carry out that process of decay. In that case now, enzymes may be found there, but not secreted by the dead cells, but secreted in the organisms of the microorganisms that may be found on the dead cell or in the dead cell. Enzymes are used to catalyze a vast number of chemical reactions at temperatures suitable for living organisms between 5 and 40 degrees centigrade. This is also very important because it is important for us to understand here that enzyme can carry out a large number of chemical reactions, but this must be done under appropriate conditions, that is, under a conducive or suitable temperature. because some of these enzymes may be affected by a certain degree of temperature. That is why the range at which most enzymes will work conveniently is between 5 and 40 degrees centigrade, because temperatures lower than 5 or greater than 40 degrees centigrade may affect the activity of enzymes. The next thing we may want to find out will be to ask ourselves, why is the shape of enzymes so important? I want all of you to think about that. Why is the shape of enzymes so important? The shape of an enzyme is very important because it has a direct effect on how it catalyzes a reaction. It has a direct effect on how it catalyzes a reaction. That is why the shape of an enzyme is so important. Because if the shape of an enzyme is affected, it will therefore affect the reaction, because the substrate may not be able to bind to it. Therefore, the shape is important because at some point it should be in conformity to that of the substrate. Why do enzymes have different shapes? This is a very interesting question. Why do enzymes have different shapes? We have to understand that enzymes catalyze different reactions, and these reactions are not similar, and they are exposed to different substrates. Therefore, the need for this question, why do enzymes have different shapes? An enzyme's shape is determined by the sequence of amino acids in its structure. Is determined by the sequence of amino acids in its structure. This is very important because amino acids are very important because they help in giving a meaning to an enzyme. The type of amino acids and its structure makes the enzyme to have a particular shape. And the bonds which are formed between the atoms of these molecules. So, the type of bonds that are formed between the atoms of these molecules determines the shape of the enzyme. Not only is the fact that the shape is made up of amino acids, but the type of bonds that link these amino acids give an idea or give the enzyme a particular shape. This now gives us an opportunity to ask the following question. Why are enzymes said to be specific? Why are enzymes said to be specific? From what we just said above, We talked about the shape of the enzyme. We just said that it is made up of amino acids and also the type of bonds that link the amino acids give us an idea of the shape of an enzyme. Now, what makes enzymes to be specific? Now, specificity is important because enzyme catalyzed reactions are not similar. They are different from each other. That is why enzymes are very specific about the type of reaction they catalyze. They are very, very specific about the type of reaction they catalyze. It therefore means that two different enzymes cannot catalyze the same reaction. Two different enzymes cannot catalyze the same reaction. Therefore, enzyme A cannot catalyze the reaction catalyzed by enzyme B. Therefore, enzyme A is specific to catalyze its own reaction, while enzyme B is specific to catalyze its own reaction. Depends on them where reaction take place. So, I want us to understand here that an enzyme molecule has points where reactions take place. Because if we are talking about the specificity of an enzyme, it therefore means that on the enzyme molecule, there are areas which we call points on which reactions normally take place. These areas or points are called active sites. These areas or points are called active sites. And this is the site where reactions occur. This is the site where reactions occur. This is the site that will normally accept a substrate. This is the site that will normally have a structure that is similar to that of the substrate, that will be able to accommodate the substrate. That is why they are said to be specific. This is where an enzyme comes into contact with the substrate. Only molecules with exactly the right shape will bind to the enzyme and react. Only molecules with exactly the right shape will bind to the enzyme and react. It therefore means that the site of an enzyme must have the same shape to that of the substrate. That is, that molecule that has to bind to that active site. Because if the active site of an enzyme has a different shape to that of the substrate, it therefore means that the substrate will not be able to bind to the active site. Therefore, no reaction will occur, or there will be no reaction in that particular case. These molecules that bind to the active site of an enzyme are called reactants or substrates. Each enzyme has its own specific substrate on which it acts. Therefore, just like what I said some time ago, enzymes are specific in their reaction. That's why we are saying here that each enzyme has its own specific substrate on which it acts. And that's why I said that enzyme A cannot act on a substrate that is destined for enzyme B. Enzyme A would act on a substrate of enzyme A, and enzyme B will act on a substrate of enzyme B. So they cannot maybe overlap with their reactions. This is then referred to as enzyme specificity. Examples here will include the following. The enzyme which act on the substrate starch, converting it to maltose, is amylase. The enzyme which acts on the substrate starch, converting it to maltose, is amylase. Therefore, the enzyme amylase cannot catalyze another reaction. It is destined to convert starch to maltose. It can never convert another molecule which is not starch. That is where specificity comes into play. The enzyme maltase will only act on maltose to convert it to 2 glucose molecules. Therefore, maltase cannot convert starch to maltose. Amylase cannot convert maltose to 2 glucose molecules. That is what we mean by specificity. When we look at this diagram, it gives us an idea of an enzyme molecule that has its active site. If you look at this depressed region, that is the active site into which the substrate has to bind, and the substrate should have the shape of this active site. Now look at the next diagram which I just come up. This is now an example of a substrate. If you look at the structure of this substrate, it is in conformity to the active site. Therefore, this substrate can conveniently bind to this enzyme to carry out a reaction. Therefore, this enzyme can carry out this reaction by combining to this substrate. We will end this lesson 1 here for today, and hoping that we continue with the next lesson for a better understanding of this particular chapter.